This is a bit of biology with Mr. Rock, and today we are going to be talking about genetic mutations. Now, this is not going to be a video about X-Men and Spider-Man, and in Hollywood you see these mutations and they give you really cool superpowers. In biology class, a mutation is going to be defined as any change in a gene or chromosome. So anytime you're changing the gene, which is the DNA, or you're changing a chromosome, that's going to be a mutation. Now, I want to be clear in saying that mutations are not always good and they are not always bad. Sometimes they're good. So modern day mutations that are kind of cool. One, there is a mutation that gives individuals increased bone density, so that means your bones are a little stronger. And there's also a mutation that gives us malaria resistance. So both of these things are really good for humans. However, there are also many bad mutations for humans. So examples would be albinism, or being albino. You have increased sensitivity to the sun due to your lack of pigment. Down syndrome is another mutation, as well as sickle cell anemia, and we're going to talk more about those in a little bit. So these are the mutations we're going to be talking about in this video, and I would say that they increase in severity. So first, there is DNA mutations. In DNA mutations, there are two of them. There are point mutations, and there are frame shift mutations. Point mutations on a scale of 1 to 5 are probably a 1, being less severe. Frame shift mutations are a bigger deal. So they are more severe and they mess up more things, or they change more things in your DNA. Chromosomal mutations are more significant and have a bigger impact than DNA mutations. So with chromosomal, there are deletions, there are insertions, and then there is this thing called non-disjunction. So deletion and insertion are pretty severe. They result in quite a bit of change. And then non-disjunction, I would argue, results in a lot of change. So kind of from the top is least impactful, and then the bottom as it goes down is more impactful as far as changes that affect our genes and eventually our traits. So starting off with the first one, point mutation. A point mutation is any time a single letter is substituted in a DNA sequence. So I think this is best illustrated via the pictures. So if you look here, I know it's a little hard to see, but that right there is a point mutation, where the original sequence read TAACT, and this T was now substituted to become a C. Same thing with to the right. It's just another example over here. This is our base sequence, and a point mutation just changes one letter. So this one letter substitution is going to affect the amino acid. And one amino acid, it's not that big of a deal. However, it does lead to some genetic diseases. An example would be sickle cell anemia, where the shape of your red blood cells are altered. So it causes bad circulation for your blood. Now, a frame shift mutation is a bigger deal, and it affects more genes and more amino acids as well. So this is where there is an insertion or deletion of a nucleotide. So we're still talking about a DNA sequence, but an insertion or a deletion of a nucleotide, and this results in a change to the way the mRNA sequence is read. So here, you got to remember DNA makes mRNA, and mRNA makes protein. Proteins are made up of amino acids. So here we have, this is like the base amino, this is the mRNA sequence, and then that's the amino acids. When we go down, we have an insertion of an A, and then that's going to mess up the amino acid sequence. So it's going to shift the letters down, and since codons are read 3 letters at a time, it's going to misread the mRNA sequence, making a new strand of proteins. So I think this is best demonstrated with this picture. I love this picture. The top is a sentence that's 3 words of 3 letters at a time. So it reads, the big boy saw the new cat eat the hot dog. And a frame shift mutation is when a single letter or multiple letters are added to shift the mRNA or the words of the sentence. So if you add letters, it becomes the big boy saw, and you add a T. Now the whole entire sentence is messed up because the letters move down 1 position. And same thing down here. If you get rid of a letter, like the letter B, it's going to move the whole entire sentence over 1, and now none of it's going to be read correctly. So those are DNA mutations. We're going to finish off with chromosomal mutations. These are where, so we're just talking about a couple of nucleotides being inserted, deleted, or substituted. Now we're talking about whole portions of a chromosome being removed or added. So a deletion, simply put, is when part of a chromosome is left out, and this occurs during meiosis. So the formation of sex cells. Here you have your sequence A, B, C, D, E, and a deletion is where part of the chromosome is left out. So the D is missing. It got removed from the chromosome. An insertion is just the opposite of a deletion. So it's when part of a chromatid or a section of a chromatid breaks off and adds on to the other side. And this results in the duplication of genes. So here we have the same sequence over again. It's A, B, C, D, E. And an insertion is when you add an additional part to that chromosome. So instead of being A, B, C, D, E, now it is A, B, C. That part is inserted, B, C, D, E. So that's an insertion. It results in a duplication of genes on the same chromosome. And once again, this is happening during crossing over in meiosis. Finally, and I think this is probably the biggest genetic mutation that we're going to talk about today, and it's called non-disjunction. You need to think back to anaphase, where chromosomes and chromatids separate from each other. So when you learned about this, the chromosomes separated evenly, but in non-disjunction, chromosomes or chromatids do not separate equally. So that is best shown. So this is metaphase. Okay, chromosomes line up in the middle. And what we learned about was, this is like proper disjunction. So you see half of it going this way and half of it going that way. But non-disjunction is when the whole chromosome is being pulled apart and it's going in only one direction. So instead of half being going left and half going right, the whole chromosome is going left. So this results in a lot of different genetic diseases, and it can happen a lot of different ways. But this is a picture of what, well, one, it's a picture of a karyogram or a karyotype. So that is a picture of somebody's chromosomes. So this is a human chromosome chart, and what you'll see is you have 2 pairs of every chromosome, and you have all the way down to 23. You see this is an XX, so this is a female. Typically, humans have 46 chromosomes, but it's boxed on this picture, and I'll box it again here. Non-disjunction results in less than 46 chromosomes or more than 46 chromosomes. So this individual has an additional 21st chromosome, and the name for Down syndrome, another name for it, is trisomy 21, and that means that somebody has 3 chromosomes instead of 2. So this individual that would have this disease actually has 47 chromosomes. So this is the result of non-disjunction, which is an uneven separation of chromatids during cell division. This has been a bit of biology with Mr. Rock. I'm signing off.